See metaphase 1. See friends, here is a metaphase 1. Here in metaphase 1 it is nothing but the homologous pairs move through the metaphase plate. See here, this is the equatorial line friends, and the kinetochore microtubule attached at the kinetochore. See, this is the kinetochore we have discussed. So, these are the microtubules friends, they attach to the kinetochore. So, they are arranged or they are arranged at the same line. See here, this is the equatorial plane friends, they are arranged in the same line. So, this is the metaphase 1. So, what is next one? Yes friends, the next one is anaphase 1. So, what you can observe here? Yes friends, here you can see that the homologous pairs move to the opposite directions. See, they move to the opposite directions. So, here they moved, but the sister chromatids remains associated at the centromere. Listen friends, here the sister chromatids remains associated at the centromere. This is the anaphase. So, during the telophase, see the next one is telophase 1. During this telophase, we can see that here the nuclear envelope disintegrates. So, the cytokinesis follows this, is called as dyad of cells. See friends, here dyad of cells, and you know that interkinesis, here interkinesis is nothing but the stage between one meiotic division to another meiotic division. So, friends, here this interkinesis follows prophase 1. See, here we have completed telophase 1, and also we can observe that here see this, there are two cells, there are two haploid cells. This is the end result of meiosis 1 friends. See here, this is the end result of meiosis 1, formation of two haploid cells, this is 1 and this is 1. These are the formation of two haploid cells. So, now we have completed meiosis 1. So, the next one is meiosis 2. Friends, meiosis 2, it is nothing but here this is the second part of meiotic division. It is called as equational division. So, this meiosis is similar to mitosis. Listen friends, this meiosis is similar to mitosis, but the genetic result is different. And also here the end result of meiosis 2 is formation of 4 haploid cells. Listen friends, the end result of meiosis 2 is formation of 4 haploid cells. So, now let us discuss the steps of meiosis. In that, first one is, see friends, here this is the meiosis 2. See, this first one is prophase 2. What happens in prophase 2? See friends, here observe carefully, this is the nuclear envelope, this one, this is the nuclear envelope. So, in prophase 2 what happens? It means the nuclear disappears and the telophase envelope again, it means it wrap up again. In the end here we can see that chromosomes are condensed. See friends, here they are condensed and they move to the polar region. See, here the centrioles move to the polar region. This is the prophase 2. Listen friends, this prophase 2 is similar to prophase 1. And also the next one is, what is the next one friends? Yes, you are right, the next one is metaphase 2. See, metaphase 2 is nothing but, here look here, observe here friends, they are arranged in the same line. See, they are arranged in the same line and the kinetochore attached to the, see these microtubules attached to the kinetochore. It is metaphase 2, and the anaphase 2 is nothing but, listen carefully friends, here anaphase 2, it is meiosis 2 also similar to meiosis 1. So, here in anaphase 2 we can see that these chromosomes are separated and they move to the opposite directions. We can observe here, but they remain constant or they remain associated at the centromere. See friends, here they remain associated at the centromere. So, the telophase is the last one. The telophase is the end one of the meiosis II. So, here we can see the cytokinesis envelope again in telophase 2 and it forms the 4 haploid daughter cells. Listen friends, it forms 4 haploid daughter cells. This is the end result of meiosis II. Listen friends, the end result of meiosis I is formation of 2 haploid cells and the end result of meiosis II is formation of 4 haploid cells. So, now let us see a live animation or a live picture of this meiosis. See friends. See, here is a animation friends, observe carefully. See here the genetic material in the nucleus. So, these are the chromosomes friends, they looks like what? Yes, they looks like threads. So, these are the chromosomes. See, observe carefully friends, are you following me? How interesting this is? Yes, see these are the chromosomes friends, these all are chromosomes. So, during the interphase friends, here look, during the interphase they condenses, it means it produces the another copy, they duplicate here. Zooming it, we can see that DNA is in the chromosomes. So, these are like beads on string. See here they are also condensed. During the prophase 1, listen carefully friends, during the prophase 1 these are the centrioles, they duplicate and move to the polar regions, they migrate to the opposite regions. See. So, here these are the chromosomes, they are condensing friends, see how they are condensed, and this is the centromere, this part centromere, and here it is the genetic recombination. Listen friends, here it is the genetic recombination, see how it is. Yes, here the nuclear envelope disintegrated and it forms the microtubules, spindle fibers friends, here they form the spindle fibers. So, observe here carefully, here the spindle fibers attached to the chromosomes at the kinetochore. During the metaphase 1, these all chromosomes are, see, observe carefully friends, here during the metaphase 1 they are arranged in the same line in the equatorial plane, and in anaphase 1 we can see the homologous chromosomes are separated. See how they are separated. See friends, here they are separated to the opposite directions. See, yes, during the in the telophase 1 we can see that 2 haploid cells are formed. See, this is one and this is the second one. These are the 2 haploid cells. This is the end result of meiosis I friends. Now let us see about meiosis II. See here also this nuclear envelope disintegrated and microtubules are formed. Here all the chromosomes comes in a same line and they move to the opposite directions. Here it also forms 4 haploid cells. See here friends, we can observe 4 haploid cells are formed. So, this here the 4 haploid cells are formed. This is the end result of meiosis II. See friends, the end result of meiosis I is formation of 4 haploid cells and the end result of meiosis II is formation of 2 haploid cells. So, we have seen the animation now. Now, we will see this. So, friends, I will ask some questions for you. Now, let us take a small break and recall what we have discussed now. See, we can see the questions here friends. What is mitosis and meiosis? We have discussed what is mitosis and meiosis. Yes friends, if the cell division occurs in other than what? Yes, in other than reproductive cells is called as mitosis, and if the cell division occurs in reproductive cells, it is called meiosis. So, then the next one is where does meiosis occurs? See friends, where does meiosis occurs? It means we can see that this meiosis occurs in eukaryotic life cycles, or it occurs in, yes friends, this meiosis occurs in where? Yes, Here the meiosis occurs in eukaryotic life cycle and in reproductive cells. So friends, here we can see that they are occurring in reproductive cells. So the next one is, what is synapsis? Listen friends, what is synapsis? You know what is synapsis? Yes, the process of pairing of homologous chromosomes is called as synapsis. So next one is, what is synaptonymal complex friends? Yes, you are absolutely right friends. Synaptonymal complex is nothing but here the synapsis or homologous chromosomes takes place and forms a complex structure. It is called synaptonymal complex. So the next one is, what is interphase? Yes, the time gap between one meiotic division to another meiotic division is called as interphase. And what is prophase? Yes, prophase is the first meiotic division friends. So here again another question, what is the steps of meiosis? See friends, this is very important. What are the steps of meiosis? Yes, in meiosis 1 we can see that prophase 1, metaphase 1, anaphase 1 and telophase 1. So in meiosis 2 we can see prophase 2, metaphase 2, anaphase 2 and telophase 2. These are the steps of meiosis. So friends, here now we will see the significance of meiosis. See friends, here these are the significance of meiosis. You know, here it also increases the genetic variability in the population of organisms from one generation to the next. So these variations, the genetic variations are very important for the process of evolution. Friends, are you following me? Listen carefully. This meiosis is the mechanism by which conservation of specific chromosomes and the number of chromosomes, see number of each species is achieved across generations. So another one we can see friends here, what is it? Yes, this is maize, we know that. So here this is paspalum, and friends the next one is brassica, see how wonderful it is. And the another one is soya bean. See friends, these are some of the examples where the breeding occurs. So here we know, and we can see here this meiosis is necessary part of life cycle of sexually reproducing animals and plants. So it helps in the, listen carefully friends, this helps in the restoring of chromosomes, the characteristics of species. So this meiosis is most essential for the completion of life cycle. Listen carefully friends, this meiosis is most essential for the completion of life cycle of plants. So recent molecular data suggests that like animals, plants possess a common set of genes. It is responsible for meiotic recombination and chromosome segregation. So friends, another one is in species, like we have seen that maize, paspalum, brassica and soya bean. So here the mutation on chromosomes pairing exploited successfully here, and in this centrosomes or this chromosomes, listen carefully friends, here the chromosomes they form cell to cell. It means the chromosomes transform cell to another cell. So here it is in the cytoplasmic connections. So it is the cytomixis. Listen carefully friends, cytomixis. So it is observed in Brassica narpus. It is observed in Brassica narpus. It is the pollen sterility. What is it friends? Pollen sterility. So we have discussed that the significance of meiosis. So here friends, you know, now let us see the animation again and get a clarify about that. So friends, here we can see the animation clearly, and now we will discuss the clearly about it. So see friends, here is a wonderful animation you have seen before. So this is the animation friends. So friends, here it is the animation. you can see these are the threads. Listen carefully friends, these are the thread like structures, they are chromosomes. So, here we also see that these are the chromosomes friends. So, during interphase they condenses, clearly observe it, here they condenses, it means they duplicate and they form another identical copy. See friends, here we can see that another identical copy. So, if we see zoom in it, we can see the DNA is present here. This is the DNA friends, and they are also condensed. These are like beads on string. During the prophase 1, the centrioles duplicate and they migrate to the opposite directions. And friends, here the chromosomes condensed, observe carefully, they are condensed and it is the centromere friends. This is the centromere, and here we can see it is the genetic recombination. Listen, this is the genetic recombination friends. Here this is the nuclear envelope, this was disintegrated and here the spindle fibers are formed. See friends, these are the centrioles, this is the spindle fibers formed, and you can see that these spindle fibers attached to the kinetochore of the chromosome. And see here friends, they are attached to the chromosome. In metaphase 1 we can see they are all arranged in the same line, it means on the equatorial plane. This is the equatorial plane friends, see. And in anaphase 1 we can see the homologous chromosomes separate. See here we can see the separation. This is a separation friends. So, here they are separated and during the telophase 1 they form 2 haploid cells. See here, these are the 2 haploid cell friends, how wonderful they are. Yes, yeah, the 2 haploid cell, this is the end result of meiosis 1. Now, we will see meiosis 2. Here also this nuclear envelope disintegrated and spindle fibers are formed. Here the chromosomes come in the same line and move to the opposite directions. See friends, at last it forms the 4 haploid cells. See how beautiful it is friends. Yes, here it formed 4 haploid cells. This is the end result of meiosis 2. So friends, now let us see a small demo. See friends, here is a small demo of meiosis which the genetic recombination occurs. So see, here they are, we think these are the chromosomes. So here they are only one, we can say it as homologous chromosome. Listen friends, here it is only one, we can say that it is homologous chromosome. So during the interphase they are duplicated. See, observe carefully friends, during the interphase they are duplicated. So these are called sister chromatids. So here we can see that these are called sister chromatids. No, this is the centromere friends. This is the centromere, here this part is called centromere. So during the prophase 1 they come together. See friends, like this, during the prophase 1 they come together and here they exchange the genetic information. We know here in prophase 1 we can see that leptotin, zygotin, pakitin, diplotin and diakinesis. So they all happen in this. So they are exchanging their genes. During the metaphase plate they are arranged in the equatorial plane. These friends are arranged in the same line. See here, they are arranged in the same line, and here we can see that during the anaphase they move to the opposite directions. See friends, they move to the opposite directions and here they form 2 haploid cells. See friends, here they all forms 2 haploid cells. This is, see, this is the end result of meiosis 1. See friends, this is one and the next one is, observe here, this is the second one. Here the 2 are formed, 2 haploid cells are formed. So now we will see about the meiosis 2. See friends, now we will see about the meiosis 2. This meiosis 2 is similar to meiosis 1. So here see, they are arranged like this. We have said that, see, they are arranged like this. And so during the metaphase plate we are saying that in prophase they are exchanging the genetic information like this friends. Here they exchange the genetic information. See, so this we can say as recombination. See friends, here this is we can say as genetic recombination. So this is the nothing but changing of chromosomes. It means changing of genes. So friends, here we can see that genetic recombination happened here like that. See, in metaphase plate they are arranged in the same line like this friends. They are arranged in the same line on the equatorial plane. During the anaphase they move to the opposite directions. Here we can see they move to the opposite direction friends, and they form 4 haploid cells. See friends, here during this they form 4 haploid cells. This is one, this is second one friends, this is third one and fourth one. These are the 4 haploid cells. This is the end result of meiosis II. So friends, now let us recall all we have discussed today. See friends, today what we have discussed, did you remember it? Listen. So we have discussed that first one is what is cell. Friends, cell is a basic structural and functional unit of a life, and this cell is having 2 basic types. They are prokaryotic and eukaryotic. Prokaryotic is nothing but, here in prokaryotic we can see that disorganized nucleus is present. Hello? Hello?
Hello.
Yes, please tell your doubt.
Good afternoon, Tony Sparrow.
Good afternoon.
Your class is so nice.
Thank you.
I am Vidya Sparrow from Suryapet.
Hi Vidya, how are you?
Fine. I would like to ask a question.
Yes, ask.
What is the main reason for this dikinesis in very thick in color?
Again please.
What is the main reason
Yes.
this dikinesis is in very thick in color?
So friends, a wonderful question. Listen here, the dikinesis cell, they don't present in the plant cell. These centrioles only present in animal cell, and the spindle fibers they are forming from the centrioles only. Friend, is it clear for you? So now let us see, we have prepared a small model friends. We can see that, see, this is the meiosis total. So here this is the prophase friends. We have seen that here we can see the spindle fibers are formed and the centrioles move to the opposite direction in prophase 1, and in the metaphase 1 we can see that, see, observe carefully friends, in metaphase here they are arranged at the same line. You can observe they are arranged the same line. During the prophase they move to the opposite direction. What? Homologous chromosomes move to the opposite directions, and the end result of meiosis is formation of 4 haploid cells. And also here see friends, this is the genetic recombination. See how the recombination takes place. So here it forms 4 haploid daughter cells. This is the process of meiosis. So now I will give a small assignment for you friends. I think you will do it and submit to your teachers. See here. Friends, you can observe, see, assignment. See here friends, I will give a small assignment. Note down please friends. First one, what are the various stages of meiotic process 1? Listen carefully friends, what are the various stages of meiotic prophase 1? So please note down this and enumerate the chromosomal events during the each stage. See friends, enumerate. Here we can see that. Please take down these questions. See friends here. Did you observe? Enumerate the chromosomal event during the when each stages. So this is the first one, and the second one is write brief note on the following. Friends, please note down this. Write brief note on the following. It is first one is synaptonymal complex. What is the first one? Yes, first one is synaptonymal complex and the second one is metaphase plate. See friends, write brief note on the following. First one is synaptonymal complex and the second one is metaphase plate. So here friends, today we have discussed many things. What are they? Cell, prokaryotic, eukaryotic. So here we can see that various plants are forming. Here the scientists are doing that various breedings for we all are depending on the plants. Due to that, to know more anything or to know more about plants, here the scientists are doing many things. So friends, this is the today's conclusion. Here this meiosis, here we can see in more stages. So here we can see that plants are also occurring in many stages. Here these plants are very useful for us. See friends, and also we can see these stages of meiosis, synapsis, synaptonymal complex, and we have discussed the prophase, metaphase, anaphase, telophase, etc. And also we have discussed this prophase, metaphase, anaphase, telophase and the steps of meiosis, cell, chromosomes, genes and how we resemble our parents. Do we resemble our parents totally? Like that many things we have discussed. So again what we have discussed? Okay, bye friends. Thank you.